Hello, welcome to this video tutorial showing how to use JContextExplorer. This is the third video in a series of 3. If you have not seen videos 1 or 2, please click the appropriate links on the screen. Alright, in the first video tutorial I explained that JContextExplorer does 3 different things. First off, it provides various ways to define genomic groupings, genomic contexts, gene neighborhoods, and then JContextExplorer provides alternative ways to perform pairwise comparisons of these defined genomic groupings. And finally, JContextExplorer provides alternative ways to assemble these pairwise comparisons into a linkage tree. So from the second tutorial, we installed this software, and so I'll go through this quickly. And now we are in the mainframe of the JContextExplorer software. Once the mainframe is opened, you should always start in the upper left-hand corner, here where it says Search Genome. And as an example, I'm going to search for the text query glycerol dehydrogenase. Alright, and JContextExplorer then searches through the information you loaded and draws a tree. In this case, the tree is not very interesting, just a list of nodes. That's because the context that we're currently working with is single gene. So JContextExplorer simply retrieves all genes that have this textual query in their annotation. So about these node names, each node name is the name of the species plus a serial number indicating the instance. So in this example, Halobacterium species NRC1-2. These nodes, you can select nodes by clicking on them. You can hold down the control key to select multiple nodes or deselect nodes. You can hold down the shift key to select all nodes between a pre-selected node and your currently selected node. You can type in the nodes you'd like to select in the bar. So for example, let's type in Halothyrax. It searches through all the node names and will select all that have this name. There they are, Halothyrax volcanii 1 and Halothyrax volcanii 2. Once you've done that, let's go ahead. You can also deselect all and select all, these buttons down here. And I'm going to go ahead and view annotations. So I have 8 selected nodes, and here are the nodes in bold, and the actual annotation that these particular genes have is here, and as you can see, within that you have this glycerol dehydrogenase string, which is the same as our query. So finally, once you have these nodes, you can actually view them. You can look at them as they occur in the genome using the Context Viewer multi-genome browser tool. And you select the nodes of interest, and you hit View Context. Now that we're in the multi-genome browser context viewer tool view, we can actually see the genes in the genomes as they occur. So each entry here is a particular genomic segment for that node. This is node NRC1-1 right there. Here it is. And we can investigate the genes by clicking on them, left-clicking. So if I left-click on our query, you see glycerol dehydrogenase. You can see other information. Left-click again for the frame to disappear. You can decide what information you want to show on click down here in gene information. And we can do that not just for a query, but for any gene. If I click outside, nothing happens. If I click within the range of a gene, then I can learn what that gene is. Okay, so we can also edit how these entries are displayed. For example, we can show the coordinates, so we can see, number one, which contig this gene occurred on. and number 2, how that sits with respect to the coordinates with these markers every 1,000 nucleotides or so. We can also, if you look down, you'll see that some of these queries occur above the line and some below. That's to represent forward or reverse strand. So if I hit strand normalize, it will recast everything to flip everything over on the forward strand. Now if I show coordinates, I'll see that for the ones where I flipped over, I've got a red flag, and the direction now indicates that transcription is occurring in this direction. Black flag, it occurs left to right, red flag, right to left. Okay, so right now, the query, in fact the entire genomic context associated with this query is just a single gene, since we did a single gene search, single gene. But we still can see the surrounding genes. So if I uncheck show surrounding, we no longer see them. I'll recheck show surrounding and now select color surrounding. We see not only with the genes, we also see them color matched. So this guy, a hypothetical protein, matched to this hypothetical protein. And this phosphoesterase is color matched to this phosphoesterase. So if you can also change the range view here. So if you can see that as a default you get 1,000 nucleotides on either side of the edge, both before and after the longest genomic grouping. But now if I update context, I've made that 2,000 before, 2,000 after. So if you're interested in examining multiple annotated elements at once, you can right-click anywhere on the frame and go to Show Legend, and I'll just do annotations, since this is an annotation search. And now I get a list of the annotations and the associated color. For example, this blue, glycerol dehydrogenase right here. And if I actually click on the entry in the legend, it will select that in the Context Viewer tool. And once again, I can control-click and shift-click just as before. And you see that many more things are selected. Finally, in that legend frame, you can also export this legend as an image. And you can export what you see here as an image. And the rule is that whatever you see, the image will show that. So if you don't want to see the surrounding things, export it like this. If you do, then like this. If you want to color them, like this. You can close this without any problem. So we can also perform multiple query searches. So here I'm going to search for either glycerol dehydrogenase or hexokinase. And now we see a tree, and the tree is segregated according to those matches that matched with glycerol dehydrogenase, and those that matched with hexokinase. Demonstrate that here. So here's your glycerol dehydrogenases, and your hexokinases. And now the glycerol dehydrogenases are selected. Hexokinase selected. Alright. So now that we've looked at some single gene entries, we want to probably do something more interesting. So we can talk about adding and removing other gene grouping schemes. So let's go to this Add Remove button right here. It's going to bring up this frame, and there are a number of options. But I'm not going to explore all of them right now. I'm just going to talk about two of the most common ones. So a really common one you might want would be a range around a query. So I'm going to call this range 3000. You should give every context set a name, and ideally one that gives you some information about what you're doing. So I'm going to call this range 3000 because I want to look at all genes that are within 3000 nucleotides before and after the center of a search query. Go ahead and click Add, and now it will appear in this list along with single gene. And I'm also going to computationally predict operons. So in microbial organisms, genes can be organized into co-transcribed sets if they are adjacent to each other on the genome. Anyway, it's a common thing that occurs in these microbial species, so we can go ahead and compute operons here. I'm going to use an intergenic operon distance threshold of 50 nucleotides. require the genes be on the same strand, and hit compute. Add that to the list, and now we see we've got 3 different context sets or gene grouping algorithms, gene grouping possibilities. Hit OK. And now if I switch to range 3000, let's go back and do a search. By the way, I should add that that original search we performed is just glycerol dehydrogenase, not this one. This one, its own window, and a new search forms a new window. So you can keep multiple searches going at once and just switch windows whenever you like. You can do things like drag these around, and you can maximize them and minimize them, and you can close them. Let's go ahead and close this one, and let's close this one too. Okay, so here is a range 3000. Go ahead and let's go ahead and submit search, and we get a tree that looks like this. So let's talk about this tree. So once again, we have the nodes, and we're using range 3000 as our context set to actually create these nodes. But now we can talk about how we want to compare these nodes to each other, so the pairwise comparisons, and then also how do we take those pairwise comparisons and build a tree out of them. So the method we're using to compare nodes to each other is called the dissimilarity metric, and here it is right here. You can switch between options, and once you do switch, just hit update tree, and it will redraw the tree for the new dissimilarity metric. Now, depending on what you want to do, you might use a different dissimilarity metric. So some of them might look a little bit strange because they're not really appropriate for this data set. A really safe one here, because now we're looking at a range of 3000, the number of genes. We'll go ahead and the common genes DICE metric. DICE metric is used to compare sets of things based on common elements. So now that we have that, we can also switch how these pairwise dissimilarities are linked together to form a tree. So this is all previously implemented in the multi-dendrograms package. You can choose amongst various common clustering methods, and you'll see how the tree changes depending on the method you use. And you can also change how the tree appears. So, for example, these nodes are very large. You might want to change the size. For all of these things, by the way, make sure to make the changes and then hit update tree. So here I'm going to color the nodes a different color. And I don't see any changes. Oh, actually, I'm sorry. This is the banner. This is the nodes. And you can make changes here. So it's also important to realize that you can right-click anywhere on the tree. So we know you can left-click to select nodes and use the annotation viewer and JContextExplorer context viewer tool. You can also right-click, and you can here are some statistics about the multi-dendrogram tree. This comes from the original package, so I recommend reading into that for more details. You can see an alternative representation of the dendrogram with some values. And once again, there's an explanation of what this means in the original multi-dendrograms package user manual. And you can save this data as the text file, either as a matrix or as a Newick tree. And once again, you can export the image that you see as a JPG, PNG, or EPS file. Okay, so as a final example, let's again look at glycerol dehydrogenase, and why don't we switch context sets to that operon set. Now, because in this case we're going to be comparing new objects or new things, we actually have to perform a new search. So here we were changing how things were connected to each other, how things were displayed, so we could just update the tree, but now we're switching, so we need to submit a new search. So let's do that. And you see things look a little different. And by the way, about this band, this is a product of the multi-dendrogram, the variable group agglomerative hierarchical clustering algorithm, which you do not see in pairwise when you do not have the variable group clustering. It's all explained in more detail in the multi-dendrograms package. But in any case, you can color that band. Let's make it yellow. Okay. So let's go ahead and hit select all, and let's take a look at these contexts. And notice that here, because I'm using operons, I have predicted that I'm looking for genes that are nearby to my query gene and are on the same strand. So in this case, this is the query, and there were 2 genes that were nearby, so are grouped together. But for this particular case, nothing is nearby, so this lives in a context set by itself. Okay, thank you for watching. This has been a brief introduction how to use the JContextExplorer tool, and I would recommend exploring the tool on your own. Thank you.